So in this video we're going to have a bit of a think about the structure of DNA and how it's replicated. So DNA is obviously the molecule that encodes genetic information. It's what chromosomes and plasmids are made of. So if we're going to understand how a cell does anything, we need to have a good understanding of what DNA is, how it works, how it's structured, and how that structure affects how it functions. So we should probably know that DNA is made up of building blocks called nucleotides. So I've drawn out a nucleotide for us already, okay? And there are three parts to the structure of the nucleotide. So the first thing we've got here, you might recognise that as a phosphate group. So we've got phosphorus and some oxygens. You then might recognise this as a sugar, and in particular it's a ribose sugar. Sorry, it's a pentose sugar, which means that it has 5 carbons in it. So some sugars, a lot of sugars have 6, this one has 5. So we've got carbon 1, 2, 3, 4, 5 there, because that's a CH2 group there. So those are the 5 carbons of the sugar. So we've got a pentose sugar. And there are 2 different versions of the pentose sugar. So you'll notice I'm missing some chemical groups on carbon 2. Okay, so on carbon 2, if you've got you could either have an OH or you can just have an H. Okay, so if you've got an OH there, then you are a ribose sugar and you'll be an RNA molecule. If you've got an H there, we would call that a deoxyribose, and you will be a DNA molecule. Okay, so that's the main difference between DNA and RNA. So it's the nucleotide. Ribose, the RNA has an OH, the DNA just has an H at that carbon 2 position. Okay. The other part of the molecule, so we've got the phosphate, we've got the sugar, and then we've got here is what we've got is called a base. Okay, so the bases are made up of carbon and nitrogen. They've usually got they're a ring structure, and there are a couple of different formats of them. And those bases are the things that you might know as the A's, T's, C's and G's. So this in this case, this is a base called adenine. So we've got a phosphate group, a sugar, and then we've got a base. So for the different nucleotides, so you probably know that in DNA, we have the nucleotides A, T, C and G. So that's adenine, thymine, cytosine and guanine. And in RNA, we have A, U, C and G. So there are 5 different nucleotides that are generally used in cells. For all of these, the phosphate and the sugar stays the same. Obviously, whether it's got that OH or an H on there determines whether it's RNA or DNA. It's just the base bit that differs between those A's, T's, C's and G's. So the sugar and the phosphate are constant, the base varies. Okay, so that's what DNA is made up of at the sort of biochemical level. So let's think about the structure. And you should know that DNA is obviously the double helix, very famous structure. Okay, so if I draw out a cartoon of that structure, okay, so we've got here we've got a strand going up and down like that. So we've got our wiggly line. Okay. And then we've got a second strand, so this is our first strand, and this is sort of going round in a spiral, which is a little bit difficult to show in 2 dimensions, but this is a spiral. You can look at 3-dimensional models of DNA structure on the internet. If you just Google DNA structure, you'll get to some nice 3D cartoons. But we need a second helix, so that's 1 molecule of DNA. So that's a DNA molecule, but we have a second DNA molecule coming in to form a double helix, so they kind of coil around each other. So there's 2 different DNA molecules makes up the double helix. Okay. So the line that I've drawn, this sort of solid line, that represents the sugar sugar phosphate backbone. So what happens is the sugars and the phosphates, they all link together into these long chains. Okay, so if I just draw this quickly, so we've got a phosphate, we've then got, I'm not showing some of the detail, but the way that the nucleotides come together is like this. So we've got a sugar phosphate backbone. So the sugars and the phosphates bind together in a very regular arrangement. Okay, and then obviously if we look at the base, then the bases are sticking out like this. Okay, so that's the actual chemical structure. This is a sort of cartoon of it. This sugar phosphate backbone is the line there. So the bases, well, what they do, if I do it on this cartoon here first, what we have is at the bases, we have what's called base pairing, okay, to that second molecule of DNA. So that is going to work of, the structure is like this. There's the ribose, and then that's going to come off there to have the phosphate, okay, and then like this, again, I'm not getting the cartoons quite right, but you can see what I'm trying to achieve. Is that you've got the second rung of sugars and phosphates. Okay, so we've got a second sugar phosphate backbone. So that's the other line on the diagram. Okay, and you will notice that I've drawn it. So on this side, it was easy to draw because the sugars were the right way around. So in this way, we're going in that direction. The way that DNA works is the other strand is going in the other direction. So you'll notice these sugars are upside down from each other. Okay, and that's given a particular notation within the biochemistry. So what we say, so let's imagine this is the very beginning of a molecule of DNA. It's going to keep going down that way infinitely. Okay, well, not infinitely, but until it gets to the end. If this is the start, we call the start the 5' end. And that's because if you look at the structure, the phosphate is attached to what's called the 5' carbon. We sometimes put these prime indications in there. So that carbon 5, so 1, 2, 3, 4, 5 carbons on the molecule has the phosphate attached to it. That phosphate is the very last thing in the molecule. That's the thing that's exposed. So we call that the 5' end because the phosphate's always attached to the 5, which means at the other end, I'll just do it. There's another phosphate. So there's infinite ones in there, but then this is the very last nucleotide in the chain. Okay, so the very last one in the chain, its phosphate will still be attached because it's in the sugar background. The thing that will be free on this one is that hydroxyl group at the end. So the phosphate attaches to the hydroxyl group, so that hydroxyl group gets lost in all of these. But at the very last one at the end, we've got a hydroxyl group, and that's referred to as the 3' end. Okay, we have the DNA is 5', and actually in terms of our direction markers, what we usually do is we say we start at the 5' end and we go towards the 3', so it's actually going in that direction. So this one, that's wrong. It's going in the other direction. So notice there is my 5' phosphate at the 5' end, and there is my hydroxyl, so that would be the 3' end. So the two strands are what we call anti-parallel. I think there's just one end in parallel. Okay, they're anti-parallel, so they're going in different directions. Okay, if I look at my structure here, so let's say that this is the 5' end here. Follow that, so that will be the 3' end, and then we've got the opposite thing here. So the 5' end with the phosphate is there, the 3' end is over there. Okay, so that's the sugar phosphate backbone. Okay. Where are the bases? Well, you can see in my cartoon here, the bases are in the middle and they form bonds with each other. So they form hydrogen bonds. Okay. And they do so because of their base pairing, which I'll get onto in a second. Okay, so on this cartoon, those are the sugar phosphate backgrounds. The bases are kind of sticking out like this. So they're sticking out from the sugar phosphate background, but they are effectively sitting between the sugar phosphate backbone. So the bases are all in the middle. Like that. Okay, so they're in the middle of the structure. So the sugar phosphate backbones are on this side, the bases are in the middle. Okay. A couple of other things to notice. So you will notice that there are two different sizes of grooves here, or sort of spaces in here. It's one of my all-time pet hates is symmetrical DNA. If you see a piece of DNA where the spacing is the same for every single one, the two things are equally attached to each other, that is wrong. That is not what DNA looks like. DNA is an asymmetric space, and it has what we call grooves. So this here on this side is what we call the major groove. So this big space is a major groove. This little space, so there's one here, there's one here, that is what we call a minor groove. Okay, and that's really important because DNA binding proteins, most of them can only bind in major grooves, because they need this big space in there to bind in. They can't bind in minor grooves, and they couldn't bind if the DNA was perfectly symmetrical. So if you see a symmetrical one, it's bad and wrong. It should be an asymmetric structure with major and minor grooves. You will notice, so per complete turn, so from this point to this point, so 2 turns of the helix, so we've got one major and one minor groove in the space, okay? That distance there, we have 10 bases per turn, okay? So you have 10 sets of base pairs per turn of the molecule. Okay. The other thing to notice is the distance between the two strands is a constant distance. So they're always the same space apart. If you rotate it in the dimensions, they're always the same. It's always the same relationship in there. And that's because, so there's, you know, again here, we've got that constant distance. And that's because of the way that base pairing works. So we said that DNA had the bases A, T, C, and G. RNA has the bases A, U, C, and G. Okay? So there are base pairing rules that are really important to understand in terms of how the structure works. Okay? So the base pairing rules are as follows. A will always pair in DNA with T, and it has 2 hydrogen bonds. So 2 bonds can form between the A and the T. C will always pair with G, and there are 3 hydrogen bonds. So the bases can't pair in any way they like. A's have to pair with T's, C's have to pair with G's. If we're thinking about a pair with RNA, then A would pair with U, and again, there will be 2 hydrogen bonds in there. Okay, so A can only pair with T in DNA, C can only pair with G in DNA, RNA we've got the U, and that helps to keep this regular structure. So there's 2 types of base pair in there, which are called purines and pyrimidines. Okay, so the purines have chemical structures where they've got 2 rings. So they would look, I'm not going to draw the structures entirely right, but a purine would look something like this. So I've got a 5-membered ring and a 6-membered ring. They've got some nitrogens and whatever. I'm not putting the nitrogen in the right place. They've got some double bonds. The important thing is that they've got 2 rings. Okay. The pyrimidines have 1 ring. So they've just got a ring structure like that. Again, there's some nitrogens in there. I'm not doing the correct structure. They've got some double bonds in there. They've got stuff projecting off it, but it's a single ring. Okay. And a purine must always pair with a pyrimidine. Okay, so we've always got 2 rings on one side and 1 ring on the other. So that can flip which way round it is, but you've always got purines bound to pyrimidines. So the purines are A and G, and the pyrimidines are T, C, U. So that's why A always pairs with T. They form 2 hydrogen bonds with each other. A is a purine, T is a pyrimidine. G pairs with C, G is a purine, C is a pyrimidine, and they can form 3 hydrogen bonds together. Okay, so that's what underpins that base pairing rule. Okay, so quite a complicated structure. There's quite a lot of bits in there, but all of them are really important for creating this shape and creating a structure that can be interacted with proteins. Okay, so I'm going to very quickly think about how we might replicate this structure. Now, DNA replication is a fantastically complicated process, and there are better videos out there on the internet that's going to show you the overall process. I'm going to give you the very much potted highlights of DNA replication. As I say, if you really want to get into the biochemistry of it, be my guest, but this is kind of the important things that you need to know. Okay. So if we're thinking about DNA double helix, we'll start with the double helix. Okay, so there's the parent DNA. So that's the original copy. Okay. In order to replicate the DNA, what we do is we unzip this entire structure. So rather than being a double helix, we split it out so the 2 strands become separated. And the enzyme that does that is an enzyme called a helicase, which unzips the DNA. Okay, so we then, rather than having the base pairs all in the middle, all being nicely bond to each other, now what we've got is we've got 2 single-stranded bits and the bases are exposed like this. Okay, so again, the solid line is the sugar phosphate backbone, and these are all the bases, the A's, T's, C's and G's poking out on either side. Okay, so the enzyme that replicates DNA is something called a DNA polymerase. Okay, so what the DNA will do, it actually doesn't start with DNA, it starts with RNA. Again, if you want to get into the biochemistry of it, there are more detailed videos out there. But what it effectively does is it starts at a point in the molecule, and it starts to set base pairs. As I say, it actually starts with a small molecule of RNA, and then it starts using DNA. So the RNA polymerase comes along, so it's an enzyme, and what it basically does is it base by base, it looks at what base it is and pairs it. So if that was an A, it's put in a T. If that was a C, it's put in a G. If that was a G, it's put in a C, so on and so forth. So base by base, it goes along and it adds the next nucleotide in the chain in this sort of progressive direction. So we've got our DNA polymerase, okay, adds the new nucleotide. So it's adding a new one of these, goes from a single molecule, gets attached to be this long chain of molecules. Okay, so DNA polymerase is doing that job. So obviously it's going, and then these can recoil in the new structure, which is fine until we come to the other side of the DNA. So there's a peculiarity of DNA polymerases, which means that they can only go 5' to 3'. They're a directional enzyme. They can't go backwards and forwards. They can only go in one direction. So in this case, it would have started with the 5', and this is the 3' end, which means because the antiparallel thing, this must be the 3', and somewhere up there, wherever it's got to, there's the 5' for this strand. Okay. This strand is easy. We can just start at the bottom, run all the way up, and basically follow the helicase, adding all the nucleotides, off we go, nice long chains. Because of this 5' to 3' weirdness, doing the other strand is a little bit complicated, because the only way that the polymerase can work is to start over here and to extend going downwards. So there's my RNA polymerase. Okay, so that's the 5, that's the 3, so that's the 5' there. Okay. So we can only start there and go. So what happens is this strand, which we call the lagging strand, gets made in lots of little bits. Okay, so we've made a little bit here, but then we've also made a little bit here. So that's the next fragment that was made by the next polymerase. Again, 5' to 3'. So we end up making these little bits, which have the lovely name of Okazaki fragments. So this only happens on what we call the lagging strand. So there's an Okazaki fragment. This strand we call the leading strand. It's fine, it doesn't need anything weird. It can just go, right, I'm starting at the beginning, add all the nucleotides. On the lagging strand, because of this weird upside down directionality thing, it doesn't work, so we have to make it in lots of little bits called Okazaki fragments. And then we have a new enzyme comes in, which is called a ligase. And basically that sticks the fragments together. Okay, so the leading strand, we make it all as one big bit, just with using a DNA polymerase. On the other side, we're still using DNA polymerases, that's still what's going on, but we have to make it in lots of small bits because of this weird that the enzyme can only go in one direction. It's not very elegant. We make it in lots of little bits. Those are little bits called Okazaki fragments, and they get stuck together by an enzyme called DNA ligase. And that ligase is really important because it's an enzyme that sticks bits of DNA together. So when we come to think about biotechnology and cloning, one of the things that we do in biotechnology is to get different bits of DNA together, stick them together. We use ligases that are naturally used in DNA replication. Okay, so as I say, if you want to get into the biochemistry of this properly, there are other videos on the internet that will give you far more detail. This is just an overview of the process. Okay, so what have we learnt? We've learnt DNA is made up of nucleotides. They've got 3 bits. They've got a phosphate, a sugar, and a base. We've learnt what the different nucleotides are in DNA and RNA and the difference between the 2 types of sugars. We've thought about the double helical structure, which is this sugar-phosphate backbone with the base pairs in the middle. We've thought about the base pairing rules, so it's how many hydrogen bonds you've got and whether you're a purine or a pyrimidine, so that forces which base bonds compare with each other, so that's the rules. It's because of their biochemical structure. And then we've briefly thought about how you might replicate this structure, so it's fundamentally it's DNA polymerases are the enzyme that does it, and we have this sort of weird leading and lagging strand model. So it's a little bit complicated to replicate it, but doing this using the base pairing rules, then we can have our second strand of DNA. So quite a bit to think about there, quite a bit of biochemistry to get your head around, but go through it as many times as you like. But hopefully that's helped you to see how the structure of DNA is so important, how the biochemistry underpins the structure and therefore underpins how it's able to encode information in the genome.